We're going to take a look at sarcomeres in this video. Sarcomeres are the functional contraction units of the myofibrils, and they consist of the actin and myosin filaments. There are thousands of these sarcomeres arranged end to end along the whole length of the myofibrils. And these sarcomeres are going to run from one Z line to the next Z line. And in this microscope view of a myofibril, we've got this dark line here, that's one Z line. And the other Z line is over here. So in between there, that would be one sarcomere. And then the next sarcomere would be, the next one over to the next Z line. And the arrangement of these sarcomeres is what gives the skeletal muscle its striated appearance. So we're going to go over the anatomy of a sarcomere. This is really important to know. This will definitely be on the exam, so make sure you know this. And let's just take a look at a picture. So we have the Z line. We have one Z line there, and we've got another Z line there. And so from one Z line to the next Z line, that is a sarcomere. And the word sarcomere, sarco is talking about flesh or muscle, and mere just means part. So that's just where the name came from, just talking about this functional unit of contraction. And these Z lines are where the thin filaments are going to attach. So you can see on either side of the Z line, you've got these thin filaments attaching. And so where we just have the thin filaments here, and we've got another one over here, these are the eye bands. And the eye bands are going to have a light appearance because they consist of just the thin filaments. And so they're not going to be real dark in appearance. And the way I remember that the eye bands are light is that the word light has an I in it. And so that's how I remember that the eye bands are light in color. All right, and then in the middle here, the length of the myosin or the thick filament, that is your A band. And the A band is going to be darker in color. So the A band will have the myosin or the thick filament and then a little bit of overlap with the thin filament as well. So it's going to be darker in color than the eye band is. So this is what's going to give us our striated appearance, because we're going to have these alternating light bands, the eye bands, and then you'll have the dark A band, which is the length of the myosin or thick filament. And then you've got another light or eye band, and then you'd have another dark A band. And so that's what's going to give you that striated appearance. Okay, and then in the middle here, we've got where we just have just the thick filament, and there's no overlap with the thin filaments at all. That's called the H zone. And then in the middle of this H zone, and kind of right in the middle of the sarcomere, that is the M line. And M just stands for middle. So those are all of the parts of the sarcomere. So I would be able to, if I have a picture, be able to pick out what the parts of the sarcomere are, be able to label those. Or if I describe something on the sarcomere, for example, saying, This part is just the thin filaments. It's light in color. It's got the Z line in the middle of it. Then that would be the I band, for example. All right, let's take a look at some pictures here. So on the microscope view on the bottom, we can see one Z line. Here's another Z line. So from Z line to Z line, that would be a sarcomere. And then you can see the I band, which is just thin filaments there, so it's lighter in color. And then the other I band will be on the other side here. And then the A band you can see is darker in color. And it's the length of the myosin, or the thick filament. And we do have a little bit of overlap here on the sides with the thin filaments with the thick filaments there. And the H zone in the middle is where there's just thick filament or myosin. And then the M line is the middle of the sarcomere. There's actually a little line there. And we can also see this on this drawing as well. You can see Z line to Z line. That would be one sarcomere. And then we've got the A band, which would be the length of the thick filament or the myosin. And then we've got I band, would just be just the thin filaments. And then we'd have another I band on the other side as well. The H zone is right in the middle of the sarcomere, and we have no overlap with the thin filaments at all. There's just thick filament there. And then right in the middle of the H zone, we've got the M line. So make sure you know the parts of the sarcomere. That would be really important to know. And then the other part, the other thing to notice about this picture, so this whole structure, this bundle of contractile proteins, this whole thing would be a myofibril. And you'd have many of these myofibrils packed into the skeletal muscle cell. And then the other thing to notice is that we've got one sarcomere from Z line to Z line. And then we have another sarcomere right next door. And then you'd have another one. And so they're just going to be lined up end to end along the whole length of the myofibril. So when the muscle is contracting, all of those sarcomeres are going to be shortening. All right, and then this is just another picture just showing again the alternating light bands, the I bands. And then we've got the dark bands, or the A bands. And then you can see the Z lines here. So Z line to Z line, you've got one sarcomere. And then you've got the H zone here in the middle and the M line right here. Okay, and then let's just take a look at the thin filaments. So they're going to be made out of 3 proteins. We have actin, tropomyosin, and troponin. And let's just take a look at a picture of the thin filaments. So the actin protein, it's a globular or round protein. So the monomer, or the little subunits of the actin, is called G-actin. The G just stands for globular, just saying that it's round. And we're just going to string these together like little beads on a string. And then we're going to take 2 of these strings of the G-actin, and the string of the G-actin is called F-actin. The F stands for fibrous, meaning that it's like a rope. And we just take 2 of these F-actin strands and twist them together, and that makes an actin strand. Okay, so that is a thin filament. And then what we're going to do is we do have these little black spots here on the G-actin. The little black spots are going to bind to the myosin heads on the thick filaments. And we're going to cover those up with another protein called tropomyosin. So tropomyosin is like a rope, and it sits right over those binding spots for the myosin heads on the thin filaments. And then there's another protein on the thin filament called troponin, and that is where calcium binds. So when calcium binds to troponin, it will lift the tropomyosin rope out of the way, and that will expose the binding sites on the thin filament. And now the myosin heads can go ahead and bind to the thin filaments, and then we can start muscle contraction, and we'll look at that a little bit later. Okay, and then this is what we just talked about with the tropomyosin and the troponin. And this is just a close-up view of the thin filament. You can see the G-actin in purple, and you can see the tropomyosin rope that's going to be covering the binding sites for the myosin heads. And then here's the troponin protein that when the calcium binds to it, it'll lift the tropomyosin rope out of the way, and that'll allow the binding sites for the myosin heads to be exposed, and then the myosin can bind, and then we can start contraction. All right, let's take a look at the thick filaments. They're made out of myosin, and let's just take a look at some pictures here. So the myosin, I think it looks like two golf clubs that have been twisted together. That's what it reminds me of. So the myosin heads stick out here, and the heads are also called cross bridges. And on the myosin heads, we have two binding sites. So we've got a binding site that will bind to the actin or the thin filaments. And then we also have a binding site for ATP. Okay, and this binding site for the ATP is actually an enzyme, and it will chop off one of the phosphate groups off of the ATP, and that'll release the energy from the ATP and provide the energy for contraction. And what we're going to do is we're going to take this myosin molecule, this golf club, and we're going to attach it to another myosin molecule, so they're going to lay end to end like that. And then we're just going to lay them down, and when we do that, so one end would be here, and the other end would be there. And then we'd have another molecule, one end there, the other end there. Okay, and then one end there, and then the other end would be there. And so we would just lay them down, and this is a 3D structure. So we're also going to have, this one here, and then that one there. And so these myosin heads would go all the way around the thick filament, except right in the middle of the thick filament, there's no myosin heads or cross bridges. Okay, so right there, you can't grab onto the thin filaments, okay, because there's no myosin heads to reach up and grab onto the thin filaments. All right, so this is how everything's going to be arranged. We've got the Z lines here on either side, and that's where we're going to attach the thin filaments to. And then the thick filaments will be in the middle here. And then you can see the myosin heads can just reach up and grab onto the thin filaments. And when they do that, then they can move and they will actually pull the thin filaments towards the middle of the sarcomere. Okay, so they'll pull the Z lines on either side towards the middle of the sarcomere, and the sarcomere will actually shorten when the sarcomere contracts. And then this picture on the bottom is a really cool electron microscope view, and you can actually see the thick filament here, and you can see the little myosin heads, the cross bridges sticking out there, and then you can see the thin filaments as well. So that's a pretty cool picture. Right, and then just one more protein that we have in the sarcomere. It's called titin, and titin is an elastic protein that is going to anchor into one of the Z lines and then run right through the middle of the thick filament and then anchor to the other side. So it's just going to anchor the thick filaments in place, and they kind of act like little springs. So when the Z lines here on either side move towards the middle of the sarcomere, and the sarcomere shortens and contracts, when the muscle relaxes, we're going to move those Z lines back to their original position. And so the titin little spring here will help push the Z lines back to their original position. So that's what the titin is doing.